I just wanted to let the kids have an opportunity to kind of show you as a board what they've been doing for this last year. This is, I think, a pretty special program for the kids to be involved with. And some of the things that they are researching and doing are things that the typical high school student wouldn't get the opportunity to do. And so with the help of a couple different professors and individuals up at Purdue, they're able to do research that's more on a freshman to sophomore college level rather than on a high school level. So I think this just is going to better help prepare them as they get ready to move on to college and think about careers as it relates to science. So Madison's going to talk a little bit about what she learned and did with her project this year. Madison.
Today I'm the teacher. What I wanted to learn with my project was that the formation of tornadoes, which we had one the other day, like 5 miles north of the airport, and my question was, does the topography of the land actually affect where they would form? There's a lot of speculation about this. There's a lot of rumors that involve tornado formation, like they're attracted to motor vehicle areas because they fly up in the air or whatever, and they're attracted to like cows. No, they're not. So what I wanted to find out was, would an area be more susceptible to tornadoes if it were flatter? And what I found out using LabVIEW, which is a grammatical programming site where I had to program in, well, it's difficult to explain. Like, there are these arrays which can plot my points, and then I just add wires everywhere, and then it ends up plotting it for me and analyzing it for me. And it was very difficult to put together, but I ended up doing it. And what I found was that in my 3 test counties that I used, which was Tippecanoe, Jackson, and Marion, 2 of which had a lot because they were probably more densely populated, and the other one I chose because it was more varied in topography, as in the eastern half had more flatland and the western half had more varied, not mountainous, but like hills. And what I found was, over here, you probably can't see it very well, but over here it centered around what would be West Lafayette. And here it centered around Indianapolis, and over here it actually centered around the flatter part of the county, as opposed to the ones with the more mountainous regions. But because there are so many variables that I can't account for, like climate change, jet stream, UHI effect, which is urban heat area, which is where heat concentrates on buildings, and which actually causes storms to the northwest of most major cities to form like right there and then go on their merry way. But there's too many variables for me to account for to make a solid hypothesis that yes, tornadoes will form and move on flatter land. But in this case, I would have to do multivariate analysis, which in some cases takes like days or weeks, and I don't know how to do that. So I'm going to end it here with my board and call it a day. And I would like to thank Mr. Banks because he helped me a lot with this and he made me not stop. And my mom for driving me up 3 hours to Purdue and back, which was bad. Anyway, project.
Okay.
I'm Brent Strala. I'm a junior. And then
I'm Olivia Malone. I'm a junior.
I'm Elizabeth Fick. I'm a sophomore.
Ethan Smiley, sophomore. I'm Tim Hersey, I'm a sophomore.
okay, Olivia Malone will be presenting her project first.
Yeah, I'm going to start you off before the more impressive projects go. Alright, so I worked with glioblastoma, which is a brain cancer, usually found on your cerebral cortex, and it's typically more common in men who are over the age of 55, but it also can happen in women, but it's a really rare cancer. Anyway, so it's stage 4, and right now there is not a reliable cure for this. They have many attempts, but typically once you are diagnosed with this cancer, you only have 14 to 15 months prognosis. It's pretty much inevitable. So my original approach to this is I obviously wanted to find a way to try and decrease the growth rates. So I found this venom called chlorotoxin, which actually comes from a scorpion, and it is known for preferably binding to glioblastoma itself. There is a protein receptor on it called matrix metalloproteinase 2 that it like specifically finds in the cell and it binds to it and it goes through the cell wall, and it can help decrease its actions of spreading out its vessels of blood. Okay, I need to explain this correctly. Okay, so whenever a tumor is in the brain, it usually takes all the blood supply. It builds up all of the brain vessels, blood vessels, and takes all the blood supply, usually killing the surrounding tissue. And so whenever the chlorotoxin binds with the glioblastoma cells, it can decrease this action and therefore killing the cell and without harming the surrounding tissue. So that was my original approach, and then I wanted to go a little deeper. So I wanted to put glioblastoma cells into a gradient and separate them towards a negative and a positive charge, and they just separate in their own ways towards which pole they want to go towards, and then take each sample from each side and culture those, which is just growing more cells, and try and find the specific ways that each different subtypes of the glioblastoma is arising within the brain, and see if we can find a specific one for each of the subtypes to decrease their specific growth within the brain. So that's a continuation of the project. I don't exactly have the results for how exactly we can stop those two subtypes, but yeah, so that's the hopes of my project that should be continued over the summer at Purdue. Yeah.
Number 2.
That's pretty much it.
So my project, it was over zooxanthella and their little symbiotic algae inside coral species and sea anemone species, and they're important. They provide for the necessary products for the coral to live. They help account for 70% of the world's oxygen, and they produce glycerol, glucose, other amino acids for the coral to actually live. And what I wanted to do was I wanted to extract the zooxanthella in a more novel way, because they have to go out, and the other methods they have to individually pick out the zooxanthella, and that's hard. It takes a little while. So what I tried doing is I tried homogenizing it, syringe homogenizing it, and that's taking the polyps, which are these little guys, taking them, cutting them up, and then you're putting them in a beaker, mixing them up, and then squeezing it through a thing countless amount of times so you get arthritis or something of some sort. And so I did that, and then I centrifuged it in a microcentrifuge, and the cells were ruptured because it was spun at too of a high velocity. So my whole thing was to hopefully separate that from the matter and tissue of the sea anemone, but they killed zooxanthella. So I'm continuing I'm wanting to continue my project into finding a more reliable way, maybe working with coral next year instead of a sea anemone. So there's my project.
So again, I'm Elizabeth. I kind of took a different path than all of the other students here. I kind of started off with a project that was, I would consider like a little bit less of a physical science, like all of these guys use like cell lines and actual organisms. And I started off kind of like a survey-based project, and I was looking to test whether or not the effect of antihistamines, which is like any allergy medication, like spring allergies, food allergies, whether or not antihistamines affected the cognitive ability of high school students to take tests and perform school-related activities. So I was looking for a way, because there's like a lot of restrictions within the guidelines for like CEPI and the organization that we do our projects through. Without administering medication to students, I was trying to find a way to collect that data. So I originally planned to just go out into the school and send out emails and flyers and talk to students and people in my classes. And if anyone was taking an antihistamine, whether or not they were now or they knew that they usually get spring allergies, I was going to try and recruit them to take a pre-test and just basic like math questions, cognitive questions, critical thinking skills, things like that. They would take that test and then I would grade it just like any other teacher would, I guess, like right or wrong. And then after they had taken the antihistamine, or that would be before. So like if they knew they were going to take it ahead of time, they'd say, okay, I'm going to take your pre-test, and then while they were taking the antihistamine, within 12 hours of actually taking the medicine, I was going to have them take a parallel test to the pre-test. So basic, like, same concepts, cognitive questions, critical thinking questions. And I was going to compare those two sets of data. But as it turned out, like a couple of months before competition, I had tried to like start collecting data. I was about to start recruiting students, but I kind of realized that it really just wasn't for me, and I had wished that I would have done like a more hard physical science, like cell lines, working with something like that. So I decided not to continue that project, and I did not. That's why I do not have a board. I didn't present that project at the science fair. But I've started a new project. I have a research block like in my high school schedule to devote time towards this. And right now I'm working on getting a project started and continuing over the summer regarding artificial sweeteners. So like any substitute, sugar substitute that gets substituted in like diet sodas, soups, really like any food. Specifically aspartame and monosodium glutamate. The professor that we all work with, he hasn't really gotten back to me. I mean, they just finished like college finals and stuff like that. So I'm trying to get a hold of him and have him like kind of guide me in a certain direction. But I'm looking to test whether or not artificial sweeteners affect brain tumors and brain cancers specifically. So I'm hoping to work with either a neuron cell line or a brain tumor or brain cancer cell line, and somehow like stimulate or put it in an environment where it's exposed to an artificial sweetener and see if that affects the cells and see if it will cause a tumor or grow cancer, or the cancer will grow at a faster rate. Because right now they've done rat studies, and the rats have shown that the cancers form and the cancers grow when exposed to high amounts of artificial sweeteners, but the FDA has said that the rat tumors were actually spontaneous and not caused by artificial sweeteners. So I'm looking forward to continuing that this summer and hopefully next year, and I just am really grateful for this opportunity and to work with Mr. Banks and Dr. Gedney. So that's my whole spiel.
My name is Ethan Smiley, and I worked with Professor Cavaluto, who's part of the plant pathology and botany department at Purdue. In my experiment, I tested the effect of yeast on like Arabidopsis seeds. And if you don't know what Arabidopsis seeds are, they're like seeds that could fit on like the tip of your pen. They're really small, and they have a short growth period, like 2 weeks. So really, it enabled me to do a bunch of tests to see how these yeasts would affect the plant growth. Yeast are found naturally on the leaves of plants. So the really cool thing about this is the Biology 2 class here inoculated yeast from plants around our community. And I got to use those yeast and test to see if they had a positive effect on the growth of these Arabidopsis seeds. And so tests like this have been done in like Asian countries and with like really exotic plants, but none like locally found in North America. So that's what really made this cool, I guess, to me. So when I did the experiment, 4 out of the 5 yeast species actually had positive effects on the growth of the Arabidopsis seeds. And I measured this through quantitative and qualitative measurements. In my pictures, there's root hairs growing down from the Arabidopsis seeds, and if they were very fibrous and dense, that was a positive effect. And if they grew more than the control, the one without the yeast species, then that was also a positive effect. And 4 out of the 5 species tested grew more than the control and were very dense and fibrous. And so the really neat thing about this is if we can isolate these yeast and put them into like a foliar spray and use it as like a biofertilizer, because chemical fertilizers can damage crops. So that's kind of the continuation of my project.
My name is Ted Hardesty. I work with Clark Kennedy like many of the other students did on my project. Please excuse my speech impediment, sometimes it comes out. My project was done on Crohn's disease, which is a autoimmune disease that affects the gastrointestinal tract. It affects over 5 million people worldwide, including an estimated 1.4 million in the U.S. My test was to find the best medication to inhibit IL-6 production. IL-6, or interleukin-6, is an interleukin or cytokine that causes a high inflammatory action in mostly the large intestine, but it occurs over the whole digestive tract. Instead, when an antigen comes into your body, the body naturally sends out cytokines to attack that antigen. But in Crohn's disease, many times the autoimmune system works almost too well, and it attacks its own cells and it attacks its own tissue, causing large amounts of inflammation, causing large amount of discomfort and pain. And there are currently many drugs out there in hopes to battle Crohn's disease, but over 65% of people still have to go through surgery, which that is not optimal. What they have to do is they have to go in and take a portion of your colon out, which is what my brother is on the verge of getting, because this is why I went after this. And so I took 3 medications that are not the main line of medications, not the first medication a doctor would tell you to give. Ciprofloxacin, which is a medication that has been found to have many beneficial effects in other diseases. Very recently, research has come out about that. And then curcumin, which is of the 3 the best known, in hopes to help Crohn's disease. It's derived from the ginger plant, ginger species, and that's commonly found in food, probably most of your pantries. And this is not the regular you would get at grocery store. It's more concentrated, more geared toward medicine. And the other one is, if I can pronounce it correctly, 3-hydroxytricyl, which came out correctly. It's more of a steroidal drug, and it has not had very much research on Crohn's disease, but it's had implications on other diseases like it, such as arthritis. And I did two different experiments inside of one. One was trying to symbolize bacteria going into the body. I used lipopolysaccharides, or LPS. They're on the outer membranes of cells, which when the LPS attaches to a cell inside the body, a T cell, it sets the cell off, or a macrophage sets the cell off to release these cytokines. So the hope is to inhibit that cytokine from going on and making more cytokines, causing a greater response. And in 2 separate treatments, one, I saturated the sample with LPS. I used the cell line JAWS2, which is a dendritic cell line derived from mice. And at the first experiment, LPS was administered into it for 4 hours, which would give it time to go into the cell membranes and proliferate the response. And then it would be administered with a certain amount of medication. And the other test, which that would symbolize the antigen already being present, so a drug coming in after somebody shows symptoms. The other one was basically the same experiment flipped on its side. And it was medication being given to the person before an attack was ever present. And the drugs were administered for an hour, and then the LPS was relevant. And my results, although they were not totally conclusive, were that 3-hydroxytrypsyl was actually the best of all three, which was very surprising to me. The sad part about it is that, like same kind of situation I had with Brent, was when the cells were centrifuged before the experiment, 3/4 of my cells were destroyed. So, although these are results that most likely are accurate to a degree, they're nowhere near the amount for you to totally count as accurate. So it would have to be more experiments, the same experiment done multiple more times to show accuracy. I hope to do something. I'm actually going to move in a separate direction this summer with my project because there's been a bacterium found only the last couple of years that they believe is actually the cause of Crohn's. It's a total switch in how they believe it's working. But, I mean, as you can believe, if this disease has been known for 100 years and we're still having 70% of our population having to have surgery, something wasn't correct. And so I believe this bacterium is what's causing it. It's a very sturdy bacterium, very hard to destroy, very antibiotic resistant. But it is accessible via multiple sources. And I believe I can get my hands on it and put it in a cell and try to destroy it. That's what my hope is. Try to destroy that bacterium. And I hope to help a lot of people that like it be successful in that endeavor.
Including your brother.
I wanted to give the kids an opportunity to present and talk to you guys so you can kind of see what they've done. This is something that's really an outgrowth of what Dr. Kaiser kind of got me hooked on several years ago when he and I went up and met with Dr. Gedney, and he talked about the biology summer camp that he put on, and Dr. Kaiser says, well, we'll send some kids up to that, and Mr. Banks will take them up with him. So I'm going, okay. But some of the things that now our kids are able to do because of this collaboration we've been able to develop with Purdue is just, in my mind, these things that they're doing, I could not do with these kids. They're things that are far and beyond what I could have ever dreamed that I could do with students. And so, you know, they've done a lot of work. They are many experts in their field. And I just hope that this whole process and program keeps going because the value that these kids see in it, you know, someone will tell you, I discovered I don't want to be a research scientist, but that's okay because they have developed an interest in something that's going to take them on to the next step at the college level. And many of you may remember Shannon Newworth. She's still up at Purdue and doing real well, and they're absolutely proud of what she's doing. And she is an example of what doing a project can be for our kids because she was able to step into a research lab as a freshman going into college. And that is not typical of what most students are able to do. And so any of our students who go through this process and do that, they're going to have those opportunities if they want to take advantage of it. And I think that's what's so unique and so great about Beech Grove City Schools and allowing us to do that. So thanks for supporting us.